Good. Well, thank you for inviting me. My fear is that this talk is going to be quite different from other talks in the series. It's about behavior. And I think what we have going for us is that everybody's interested in behavior. I understand most of the talks about physics and chemistry. But I think, as parents and understanding ourselves, behavior is kind of at the forefront of that. Let's see. This is okay. So suppose I start by saying it sounds like I'm going to say step right up and sell you some snake oil. But I have a new set of predictors of behavioral traits. They're going to transform psychology and psychiatry. They predict more variance than any other measure. And they're 100% reliable, unlike most of the measures we have in the behavioral sciences. They're unbiased. You can't fake it. It's not affected by your anxiety. These are predictions that are causal, unlike just about any other predictions we have in the behavioral sciences, like the correlation between brain and behavior isn't necessarily causal from brain to behavior. Increasingly, we think behavior is causing changes in the brain. And it predicts from birth just as well as it does in adulthood, and it costs less than 100 pounds. Well, you'd say, okay, this is crazy. This is sort of like, the pop psychology section in the bookstore. But this is actually what I'm going to talk about, and that is what we call polygenic scores. So these are all predictions based on DNA, and this is really happening now. So I think there really is a revolution, and it's already affecting a lot of behavioral research. So to step back a little bit, I've been doing work in the area of behavioral genetics for 40 years, and in light of the replication crisis, I'm sure you've had discussion about that in science, I thought I would put together a paper saying that I think some of the most important things we know about behavior come from genetic research. And so this paper discusses those findings, but the first law of behavioral genetics, as it's called, is that all psychological traits show significant and substantial genetic influence. And this is rather amazing because 20, 30 years ago, it was hard to even suggest that genetics might be important. Things have changed so much that there's now a general challenge issued to find any trait, that is, any differences between people that show no genetic influence. This is so well established that it's no longer interesting to do studies that show yet another trait has substantial genetic influence. The way the science is going is to try and find the genes. So that's what I'm going to be talking about today, and that's why I call this the DNA revolution. I'll give you some basics as to why I'm saying this about genetics and genomics and the technique of genome-wide association, and especially something that's just happened in the last year or two, these polygenic scores. And then the last part of my talk will be about applications. So it's a bit provocative, my talk, and I hope it'll lead to some good discussion afterwards. For those of you who are in physics and chemistry, probably a lot of this is unfamiliar to you, but there's lots of places you can read about everything I'm talking about, including my textbook. One thing that I find is really important is to make sure that people understand the difference between means, averages of a population, and variance, which is individual differences. So that's the hardest part of understanding what I'm talking about. We're not talking about averages, like human averages, like why is the human species a natural language user? Why do mammals have attachment bonds between infants and offspring? We're talking about differences between people. There are different perspectives, levels of analysis in all of science, and a lot of science is about this, what I call normative, or dealing with universals. For example, I mentioned, why is man a natural language user? Why do children, on average, learn to use two-word sentences by the age of 18 months? That's important, and it's also important genetically, because as I'll say, of our 3 billion base pairs of DNA, over 99% of all those bases are identical for all of us. So there's a lot of common DNA, and a lot of that goes back to not just mammals, but to bacteria. So this is a very useful perspective, but here's the perspective we're going to focus on. Why are people different? So we're natural language users, but why do some children, why are they so delayed in learning language, like Einstein not using words till he was 3? So why are some children reading disabled and others not? And the thing about these perspectives is they're not right or wrong, but they're just different. And the causes of one are not necessarily the causes of others. Like man could be a natural language user, and I assume most people would guess that's due to evolutionary adaptation. But that doesn't mean genetics is also involved in individual differences. There's no necessary connection between the two levels. So the most important thing is we know when we're talking about one level or the other, and ultimately we want to understand behavior at all these levels. But this is the level I'll be focusing on today, looking not just for means, but for the variation, and the statistic is called variance, which describes the extent to which people are distributed. And this is the normal bell-shaped curve. And in between, there are these other perspectives, like there's thousands of single-gene rare disorders, and they're very important, but they're very rare. And the way they're viewed is that everyone's basically the same, except there's these rogue mutations that make people very different. And that's true for thousands of single-gene disorders, like Huntington's disease on chromosome 4, at the top of chromosome 4. You only get, it's necessary and sufficient for the development of Huntington's disease. I hope I didn't say Alzheimer's, it was Huntington's disease. And necessary in the sense that you only get Huntington's disease if you have that particular form of the gene. And it's sufficient that if you have that form of the gene, you will get Huntington's and you will die from it, regardless of your other genes, your environment, anything else, unless something kills you before that. So there are rare single genes, but they don't contribute much to variance in the population because they are so rare. So they're dramatic, they're important, this perspective is also useful. But mostly what we're dealing with in psychology and psychiatry are what we call common disorders. They're not due to single genes. And what they are due to is many genes of small effect, and that's what I'll be talking about. And that also applies to this normal variation. So different perspectives, not right or wrong, just more or less useful for different reasons. By genetics, I'm talking about a very narrow definition of genetics, inherited differences in DNA sequence. When we talk about heritability, the extent to which these differences cause differences in traits, like schizophrenia or reading disability. And it's very narrow in the sense there's a lot of DNA effects, like most cancer involves DNA, but it's not inherited. So if I get skin cancer here because I'm a sun worshiper, it's not something I inherited, it's a mutation in the DNA in my skin cell that won't be transferred to my offspring. So it's a narrow definition of genetics. By genomics, I'm sure you all know, we're talking about the 3 billion base pairs of DNA that were sequenced first in the early 2000s. We talk about the 3 billion base pairs of DNA chopped up into 23 pairs of chromosomes, and we actually have 6 billion base pairs because you get one set of chromosomes from your mother and one from your father. Now, as I said, 99% of all the sequence in our 3 billion base pairs of DNA are identical for all of us. But that means 1% is different, and you have 3 billion, that means there's a lot of differences. The most common difference in the genome is called a single nucleotide polymorphism, SNP. And that's because it's a single base of DNA, a rung in the spiral, in the sort of rope ladder, twisted rope ladder of DNA. So it's a single nucleotide polymorphism. There are, it's hard to put a number on this, but if you say SNPs that are in a 1% frequency or greater, there's maybe 80 million in the world, but any one population has maybe 8 million, and any one person maybe has 4 million. So there's a lot of these DNA variants. And even though 99% of all our DNA is the same, the reason we're interested in the differences is it takes differences to make differences. So if we're trying to explain why are people different, why are some people schizophrenic and others not, why are some alcoholic and others not, or reading disabled, we're talking about what makes them different. So we're talking about these inherited differences in DNA sequence. And to find the extent to which DNA differences are responsible for heritability of a trait, you first have to get DNA, then you have to genotype it, and then you have to ask whether or not these DNA differences are associated with the trait. So getting DNA now, you can get it from any cell. You know, it's amazing to think we start life as a single cell and we end up with our trillions of cells, all of which have the same DNA. So you could get DNA from any cell. MI5 could get it from this cup I just had my lip on, because your cells of inside your mouth get sloughed off. All you need is one cell, and you can get a DNA and amplify that DNA so that you can genotype it. Blood is most normally used if you have access to patients in hospitals. But for these large population samples, saliva is being used. And it's because it picks up sloughed off cells from inside your mouth. So this is Origin, and it's, like in America, a very widely used website is 23andMe, and also these ancestry websites. And this is typically the way they collect DNA. Extracting the DNA is easy. You just spin it down, and in the bottom of the tube is DNA, which is a very dense molecule. So I want to make sure you understand what a SNP is, because it's the essence of all of this work. And as I say, it's a single nucleotide-based polymorphism. So this is the double helix of DNA. And the base codes are written in this 4-letter alphabet, A, C, G, and T. And one strand is coding, because C's only bind with G's and T's only bind with A. So this one strand is the coding strand, and the other strand is the complement of it. So wherever there's a C here, there's a G here, T, A, T, C, G. Now, this is one chromosome, say. The double helix is one chromosome. Let's look at another chromosome, and 99% of the bases will have the same sequence. But there's one sequence, one base pair that differs. This chromosome has a C, this chromosome has a T. Those are called alleles. So this SNP consists of two alleles, C and T. They're always just two alleles. It's a single base pair, and all you can have are these 4 nucleotide bases there. Now, we inherit 2 genomes, 2 chromosomes, of the 23 pairs of chromosomes. So we inherit alleles 2 at a time. That's why we call it genotypes. So this might not be 2 chromosomes. This could be one individual's 2 chromosomes, in which case this person would happen to be CT, one C allele, one T allele. You could be CC or TT. So that's what a SNP is. Here's a famous one that shows you how we look for an association between a DNA variant and behavior. This is a very famous one in science 10 years ago, and it's an association with body mass index, height corrected for weight. And this is body weight standardized for a mean of 0 in the population. And what this is showing is that people with one A allele are heavier, actually 3 pounds heavier than people with TT alleles on average, and people with AA alleles are again 3 pounds heavier. So the difference between the TT and AA genotype is 6 pounds. That's an association. It's like a correlation between genotype and phenotype, measured trait. So it's saying the more A alleles you have, the heavier you are. So that's an association, and we've been doing association studies forever in genetics, but what's new in the last 10 years is something called genome-wide association. Prior to that, studies involved candidate gene studies. So when I first tried to find genes for learning abilities and disabilities, all you could do at the time was to take your chances, pick some genes, dopamine genes, neurotransmitter genes that are active in the brain. You think they have something to do with what you're studying, but the problem is 75% of all genes are expressed in the brain, so you could tell a story about just anything, and that was one of the problems. But it was very expensive to do genotyping, though. If you wanted to do 1,000 subjects, say, each SNP has to be genotyped separately. And so it was an expensive, slow process, so you could only do a few candidate genes. And genetics contributed its share to the replication crisis of science with these thousands of candidate gene studies. They do not replicate. For all the same reasons that we have the replication process, things like chasing p-values, that sort of thing. I can talk about that more later if people are interested. But what's changed then in the last 10 years is genome-wide association. It's the opposite of a candidate gene approach. Instead of saying, well, I think this gene or that gene is related to my trait, like serotonin was studied in relation to depression, and dopamine genes were studied in relation to attention deficit disorder. Genome-wide association is atheoretical. You simultaneously assess DNA differences across the genome for association with the trait. So you look across all 3 billion base pairs of DNA, And if you could sequence it, you could see that you would get every single difference between this person and a reference sample. Now, that's the way it's going to go, sequence the whole genome. But for now, we've been using something called a SNP chip. And it's a little difficult to understand, but I mainly want you to understand that genome-wide association doesn't just look at a few SNPs, it looks at hundreds of thousands, millions of SNPs spaced systematically across the genome so that you actually tag all the DNA, most of the DNA variation, without having to sequence all 3 billion base pairs of DNA. So that's this technique, and it involves SNP chips, which uses the same approach that's always been used to identify single nucleotide polymorphisms, but it does it at a massive parallel level. This involves hybridization. You have a probe, and you denature your DNA. You get your DNA, you get saliva, extract DNA, denature it. By raising the temperature, you make the double-stranded DNA split apart. And when it splits apart, you cut it, and you look for whether or not your single-stranded DNA finds a match with this probe. And this will be a unique sequence of DNA, so that if you hybridize, single-stranded DNA doesn't like that. They want to find their mate. So they will hybridize if they have the proper sequence. Because we're looking for a C SNP here, a polymorphism, if you have a G at that point, you will hybridize with it. So we can say, you've hybridized here. If we put a fluorescence tag on this, we'd see that your DNA hybridized so that you have a G at that spot. If instead you had a T, it wouldn't hybridize because C wants to go with G. And it won't hybridize, it'll repel if it has a T. So it's the same technique that's always been used, but what's different about genome-wide association is on a chip the size of a postage stamp, you have hundreds of thousands of these probes so that you can assess SNPs across the whole genome with just a little bit of DNA, and very quickly and now very inexpensively. And so this is a chip, and this chip consists of hundreds of thousands of these probes, and your DNA is put a fluorescent tag on it so that if you hybridize, it's bright. If you have two Gs, it'd be very bright. If you're heterozygous, GT, it'd just be green rather than yellow. And if you're TT, you'd be dark. And a laser reads that then. So it's very quick, efficient, reliable. It really works, and because it's so cheap, it's made this science, unlike whole genome sequencing, it's made it available for everybody. I mean, everyone can do this in their lab. So this was named Breakthrough of the Year in 2007, and in just 10 years, genome-wide association has identified 10,000 robust associations with hundreds of complex traits and common disorders. This is a great paper that just came out a couple months ago by Peter Vischer in the American Journal of Human Genetics, and if you're interested in this stuff, it's really very accessible and well worth reading. This is just what genome-wide associations results look like. So you can't pick up any life science journal without seeing this. It's called a Manhattan plot. And this is for height with a very large sample so that you can detect small effects. And it's called a Manhattan plot because of these spikes, which are supposed to look like the Manhattan skyline. And what they consist of are the significant areas of the chromosome. So down here we have 22 pairs, the sex chromosome isn't here. And this line is the line of genome-wide significance. Because you have so many tests, you know, the multiple testing problem, you have to correct the probability values for the number of tests. And this is an unimaginably stringent correction for 1 million tests, so that you need not the typical P of 0.05, you know, probability level of 5%. It's P of 0.05 with 6 zeros in front of that, 5 times 10 to the minus 8. And these are the regions then where there's a bunch of SNPs that are associated with the trait. They have very, very low p-values. So this is the line of genome-wide significance, and all of these SNPs are significantly associated, having corrected for genome-wide significance. Now, the reason they're in these skyscrapers is that if two SNPs are close together on a chromosome, the chances are you'll inherit them together. It's called linkage disequilibrium. So you expect to find, if there's a strong hit, that other SNPs right next to it will also show an association with the trait. So down here you have, in this case, millions of SNPs that aren't associated, but then these spikes lead to the conclusion, and this is fairly old now, it's 3 years old, of over 400 genome-wide significant results. And these really do replicate in other studies. So that was the big finding. This works. And as that previous slide showed, 10,000 traits have been shown to be reliably defined genes using this genome-wide association approach. But the other finding that people at first kind of slipped under the carpet a bit was that the average effects are incredibly small. This is 0.05%, 0.0005 of the variance, of the differences between people. An incredibly tiny effect. And that's why you needed these large samples before you can begin to detect these effects. The average effect, say if you take the top thousands of associations, is incredibly small. And to detect effects this large, you need samples of many hundreds of thousands. So the conclusion from work in height and medicine, all the life sciences, physiological traits, wherever these GWAS studies are being done, and behavior as well. Behavior is just as heritable as a lot of these other traits. The conclusion from GWAS studies is that many genes of very small effect size must be responsible for heritability. We can actually say now there are no effects, no DNA polymorphisms that account for more than 1% of the variance in the population. That one I showed you with FTO and BMI, that SNP, that AT SNP, it's a big effect, right? From 3 extra pounds for every A allele. That is one of the biggest effects found, but it still accounts for less than 1% of the variance in body mass index. So the average effect sizes are very small, and if they were, my guess is 0.005%, that would mean that if the heritability of traits is 50%, that means the differences between people 50% of the differences can be ascribed to DNA differences. That would mean we need 10,000 SNPs to account for it. And that means we need huge samples, several 100,000 individuals to pick up small effects like that. And so that's what people have been up to in the last few years. Sorry, that should say 2017. A paper just came out in the American Journal of Psychiatry from the Psychiatric Genomics Consortium, which is a brilliant consortium. To get these huge samples, like with psychiatry, they started with 60,000, and they're now up to 100,000 cases of individuals diagnosed with schizophrenia, where they have the DNA and they've done the genotyping on them. Because the first study was done by the Wellcome Trust in 2007, and it was an amazing study, and they studied 7 different diseases, only one psychiatric one, and that was bipolar disorder. And it was considered amazing at the time. They had 2,000 cases for each of the 7 disorders and 2,000 controls. They're looking for allele frequency differences between those groups. And they didn't find much, and that's when they began to realize that the effect sizes are much smaller and that what we need are much larger samples to do that. So the success has come in the last year with schizophrenia with 60,000 cases, and they're about to publish on 100,000 cases. Bipolar, 20,000 to 30,000 cases to compare to controls. Major depressive disorder is the most amazing. Last year, a paper was published with about 80,000 cases, but now there's a paper in the works for 130,000 cases. For the childhood disorders that I'm particularly interested in, there are about 10,000 cases, which just isn't enough to pick up these small effects. But they're funded now, PGC, to get to 40,000 cases for each of those. Now, these are case control studies because people believe in these diagnoses that you're schizophrenic or not, and what you have to do is diagnose it properly. I don't believe in that, and I'll come to that in a minute. I've been studying dimensions, quantitative traits, like particularly cognitive abilities and disabilities. But the one that's going to hit the news early next year is a GWAS study of 1.1 million people. And if I have time later, I'll come to the data for this, but they're now explaining, well, I'll come to that. It's not until they got to The latest paper published last summer was on about 300,000 people, and they started to make progress there, getting something like 200 significant hits, as they're called, genome regions that are correlated, say, with years of education. But now, with the new study, it's over 1,200 genome-wide significant hits. And you might ask, well, why years of education? Well, it does actually predict a lot of things in society, like learning ability. It correlates about 0.5 with learning ability. But the reason it's been used in these GWAS studies is every GWAS study ever done asks people how many years of education they've had. It's a demographic characteristic. So that's why they've been able to get the huge Ns, which demonstrate that you need those large Ns to scoop up these tiny effects. It's like we thought we were going for gold nuggets, panning for gold. But it's not nuggets, it's gold dust. And to get that gold dust, you gotta scoop up a lot of the dirt along with it. And so it's been a real revolution to realize how small the largest effects must be. No one ever anticipated they'd be that small. So I'm sure the chemists and physicists in the room, what good are these tiny effects? And if you're interested in a bottom-up molecular genetic reductionistic approach from genes to brain to behavior, it's gonna be tough. It's tough enough if you had a single gene to figure out brain behavior pathways, because it's not simple, direct lines from genes to proteins to brain to behavior. But if you're talking about effects that account for 0.005% of the variance, it's hard, and that there are tens of thousands of those involved in any trait. It's hard to imagine how you're gonna make much progress with a bottom-up approach. But being a behavioral scientist, I'm interested in using these in a top-down approach to predict behavior. And the neat thing about DNA is that although we want to know about everything, epigenetics and RNA transcription and how they work in proteins and in the brain, with inherited DNA sequence, you can skip everything and jump to behavior. You can predict behavior with inherited DNA sequence. So that's what we're doing. And the way we do that is through polygenic scores. So I've told you a bit about genetics and genomics, a bit about genome-wide association. But what this is building up to are these polygenic scores that take advantage of many tiny effects. So it's just like creating a scale. You know, if we have a personality test or a reading test, you don't have one item. It's not a very reliable predictor. You have multiple items, and you put them together in a composite. You aggregate individual items, and this is just the same thing, but instead of aggregating items, we're aggregating SNPs. So you can get a composite of SNPs, just adding them up, and that's what we're calling a polygenic score, multiple gene score. But you can do a little bit better if you weight them by their association with the trait. So here's a concrete example of constructing a polygenic score for just one individual for just 10 SNPs. Okay, so like an item on a test, you have to get it in the right direction. If I'm doing a shyness questionnaire, you reverse the items. You say, I like to go to parties. And then you say, I'm anxious when I meet people. So you have to get a shyness score, you have to reverse those items. And that's what you have to do with SNPs, because it's arbitrary which SNP is which, but you call the one that contributes to the trait positively, call it the increasing allele. Like in the AT SNP for body mass index that I showed you, the A is the increasing allele. So let's say for SNP1, T is the increasing allele. So this one individual has one T, so we give him a score of 1. This next SNP is a C, he doesn't have any of those, 0. This one's an A, has 2 of those, that's a 2. So if you add those up, that could be the genotypic score of 9. With 10 SNPs and 2 alleles, you'd have a possible score of 0 to 20. So the 9 is kind of in the middle. But you can do better than that by weighting this score by the correlation with the trait, by its effect size. So the first SNP correlates 5 times as much as these SNPs. So just as you would do in constructing a psychometric score, like a personality scale, you get a better, more predictive score if you weight this score by the correlation. So this SNP counts 5 times as much as these SNPs in creating this weighted genotypic score. If you just add that up, you get a score of 0.023, which isn't interpretable in itself, but when you put that into a population, you'll see that this score is also about average. But it's more predictive. So that's what we're talking about as polygenic scores. And do you remember I said the biggest SNP in the GWAS for height only accounted for 0.05% of the variance? But if you take the top SNPs, you can account for 17% of the variance in height in an independent sample. So that's what it's about. We're trying to use DNA alone to predict traits. And predicting 17% of the variance is a lot. And in the behavioral sciences, this is for height, but you'll see we're getting there in the behavioral sciences as well. So when I showed you before about the major GWAS studies of behavioral science, for schizophrenia, we can now explain 7%, it's called of the liability. It's because it's not variance, you've got cases and controls, you have to talk about the liability. And it's harder to present that. But you could say people in the top decile for the polygenic score for schizophrenia, derived from that GWAS study I showed you, the top decile is 15% more likely to be schizophrenic than the bottom decile. And you can translate that to variance in a population, liability, but it's a tricky business. But we talk about 7% of the variance in liability, whereas 50% or so of the liability is heritable. Bipolar depression, 3%, major depressive order, 2%. But what's really taken off are cognitive abilities. Like that study that's going to be coming out on educational attainment is explaining over 10% of the variance. We can take those polygenic scores and predict educational achievement, like GCSE scores, and explain more than 10% of the variance in GCSE scores. And also with general cognitive ability, we can explain more than 10% of the variance, 5% of the variance in reading. The one area, I showed you that they have GWAS studies of 300,000, haven't been so successful, explaining only 1% of the variance, and I don't know what that's about. These are self-report personality questionnaires, maybe that's part of it. The heritability from twin and adoption studies is quite high, though. Okay, now the other thing is that bigger and better polygenic scores are on the way. There's this issue of missing heritability, and that is that there's a big gap between a polygenic score and the known heritability. For example, most behavioral traits show about 50% heritability in twin studies. This is where you compare identical twins and non-identical twins. Identical twins are like clones, they're genetically identical. Fraternal twins, non-identical twins, are like any brother and sister, 50% similar genetically. So using that method, you find about 50% heritability, but we're explaining only 10% of the variance. And missing heritability is that gap between how much we can explain with identified SNPs in a polygenic score and heritability. and for all traits in the life sciences, there's this big missing heritability. And part of the reason is that GWAS studies and polygenic scores are limited to the SNPs on chips. And the SNPs on chips are common SNPs. Most SNPs are not common. So whole genome sequencing will probably solve some of that problem. I won't spend the time talking about that, but there's a lot of work throughout the life sciences being done on missing heritability, and there's lots of approaches to dealing with it. But the idea is we've done amazingly well with one hand tied up behind our back. That is, we can only detect about half of the heritability given the current SNPs and their use of common SNPs rather than the rare SNPs. So it's going to get bigger and better, especially with whole genome sequencing. So this is the reason why I'm really excited about polygenic scores, which have just come up in the last year or two in the behavioral sciences. They really do predict more variance than other trait measures. I mean, even take height. Do you know how much of the variance in adult height you can explain with infant height? Very little, 2%, something like that. And here you're explaining 17% of the variance with a polygenic score that predicts just as well at birth as it does in adulthood. They're completely reliable, these SNP chip genotypes. They're unbiased, of course, and they are causal in the sense that, in this very narrow sense, People get upset. I know causality is a big deal for people, but I do, in this very narrow sense, there's no reverse causation. So when you correlate DNA variation, like a polygenic score, with behavior, there's nothing in the behavior, the environment, the brain that changes the inherited DNA sequence. So there isn't that backward causation. Whereas with brain behavior correlations, I think increasingly the causation is backward. It's from behavior to the brain. I don't think reading disabled kids have brains that are structurally or functionally different as a causal mechanism. I think if kids don't read or they don't read properly, their brain is different because of that. Okay, and then it is, as research goes, relatively cheap to do these. The practical reasons for using this in the behavioral sciences is that you don't need special samples like twins or adoptees. You know, for 40 years I've been trying to get psychologists to get interested in genetics, but it's a big ask because, you know, it's hard to get twins. 1% of the population are twins, and it's increasingly hard to get adoptees, easily placed children adopted at birth. You need, the thing about the DNA revolution is you need huge samples, monstrous samples to do GWAS, but you don't need large samples to use polygenic scores. So if you have a polygenic score that explains 10% of the variance, as we do for cognitive abilities, disabilities, you only need an N of 60 to be able to detect that effect with 80% power. So you can use these polygenic scores in any research. And that's what's going to make the difference. That's what is making the difference about the DNA revolution. So let me talk a bit about these applications. I'll end by talking about research applications and some concrete examples of our research. And I do really believe we're going to transform clinical psychology and psychiatry in very big ways. But I'll begin by talking about the use of polygenic scores to understand ourselves and our children. Because in part, I think the personal genomics revolution is going to be driven by people. And already people have paid $200 to have 23andMe do their genotyping, and Ancestry is also incredibly popular. But the genotyping they do is single gene genotyping, you know, saying, do you have this particular genetic disorder or not? But where they're all going to move in the next year is towards these polygenic scores. 23andMe now is using a polygenic score for obesity, body mass index. And I know this is what gets into the ethical issues, and this is, I'm sure, will lead to a lot of discussion. But I thought to make it concrete, I'd take DNA from this guy, spitting into a tube, extracting DNA, putting it on a chip, getting my, in this case, several million SNPs, and then for all the polygenic scores, well, I'll give you samples of them, we're going to construct my polygenic score and relate it to this English population that I have, representative sample. So the question is, like for height, I'm very tall, but where's my polygenic score going to be? And, you know, I am very tall, and my polygenic score is at the 90th percentile. Now you say, but you don't need that to see I'm tall. But you wouldn't have been able to tell I'm going to be so tall at birth, for example. And 90th percentile isn't the 99th percentile. There's a lot of error here. And this is what's important to understand. This is variance again, but it's covariance. This is a scatter plot in this population I'm using, the UK population, of the polygenic score correlated with actual height. And this is in percentiles, just so that they are kind of even. Now you can see there's a relationship, right? You know, the higher your polygenic score, the taller you are on average. But with a correlation of 0.41, and you need a square correlation to get the variance explained, that's where we get the 17% of the variance can be explained by polygenic scores. That is, you can predict people's height, 17% of that variance with the polygenic score. But there's a lot of people, there's a lot of error in that estimate. Like there's me at the 99th percentile of height, but only the 90th percentile of a polygenic score. There's bigger outliers. Here's the two of the tallest people in my sample, but they actually have polygenic scores below the mean. So it's not completely accurate, but this is nothing specific about DNA. This is just statistics and correlations. And this is a correlation of 0.4. The stronger the correlation, the better prediction. But it's never going to be a correlation of 1 because heritability of these things is nothing like 1. So we're always going to be faced with this. And then some people say, well, then the polygenic scores aren't useful. Well, nothing we use for prediction is 100% accurate. You use the best predictors you have, and in this case, these are some of the strongest predictors we have. They're causal, and you can predict from birth. But you need to be aware of this issue of variance. Now, if you put that previous slide, you chop it up into deciles. So you take all the polygenic scores and you divide it into units, 10% chunks from lowest to highest. That's what these deciles are. So here's the top decile, there's the bottom decile. And then you simply look at the mean height in those deciles. And you can see the association much more clearly. You could sort of see it on the previous slide, but this makes it much clearer. And it also makes it clear that at the extreme, despite all this variability, there's quite big differences. The lowest decile for height polygenic score is at the 25th percentile of height, whereas the top decile is at the 75th percentile of height. But one more point about this is that this is that previous slide, bottom and top decile, but instead of just looking at the mean, here's the same means I showed you, but instead of just looking at the means of the lowest decile, looking at their height, and the highest decile, we're looking at their distribution. And you can see that there's 50% overlap in these distributions. So it's not at all, there's a big error in predicting individuals, but you can still make predictions as long as you're aware of that. So I'll give you some more, and because I'm just one person, these are like anecdotes. These aren't proof of anything, but this is why it's going to be interesting for people. So this is a bit confessional. I haven't done this, and I'm a little embarrassed to do this, but I am at the borderline of obese. 30 is like overweight. And this is the body mass index 30, but that's 114 kilograms at 6'5". So I'm at the 60th percentile for weight. So what's my polygenic score for weight? And it turns out it's very high, 94th percentile. Now this makes the point where people say, well, this is so dangerous knowing you're polygenic, it's like genetic fortune telling. Well, in my case, I look at that, and some people might say, oh, well, I'll give up dieting, I'll just be fat. But for me, it's actually very motivating to say, actually, I'm only at the 60th percentile. And it's also motivating to realize that it's a constant battle for me. Some people, weight is highly heritable, 60% heritable. People don't really recognize that. And it's important to recognize and respect those differences. Like me, I deserve your sympathy. That I put on weight very easily, and it's very hard for me to get rid of weight. Psychologists study two mechanisms. It's not much to do with energy balance, but psychological mechanisms that I know apply to me totally, and that's satiety, feeling full. I eat everything on the table. People, how could you eat that? You must be full. I don't know, I just keep eating. And then the other bit, sensitivity to food cues. If I smell delicious food, it's just so hard for me to resist it. I can't have the stuff in the house, or I just eat it. So you could say, well, that's a matter of willpower. But the thing is, it's a lot harder for me than it is for many of you. And so I realize I've got a constant battle. It's not like I finally lose weight and there, I'm done. It's going to be a battle. So I think that's one of the ways in which these polygenic scores can be useful, to look at, say, discrepancies between polygenic scores and actual weight. And as I say, we can also predict from birth, which is important in case of the obesity epidemic, that everyone recognizes whatever you're going to do, you have to do it very early in life. And also it's a causal prediction. So there's over 200 medical polygenic scores available. And although my talk's about behavior, I partly want to show you why it's exciting to think about polygenic scores. So this is that population I'm comparing my results to, right? And so I'm giving them polygenic scores created the same way as my polygenic score. So my polygenic score for resting heart rate is just at average. And inflammatory bowel disease, Crohn's disease, it's a bit above average. Lung cancer, that's getting up there a bit. I never smoked, I never liked the sense of smoking, but if I had known this early in life, this might have been another impetus not to smoke, because I'd be more at risk for lung cancer at an individual level. My sibling could actually have a much lower risk because first-degree relatives only share 50% of their DNA differences. Migraine, again, this is anecdotal, this is just one person, but these are predictive, 10% of, say, the liability. And I used to have migraines as I was an adolescent, and I get them fewer and fewer times. I can get a migraine anytime I want just by spending 10 hours staring at my computer screen. And I see the signs for it, things start getting that visual impairment. So it's kind of interesting that, it's something I should watch out for, because it's not nice to get migraines. And the more you get them, it seems like the more you get them, So here's insulin sensitivity is actually a good one. It's protective against diabetes. And then here's the one that really struck me, a very high genetic risk for rheumatoid arthritis, which is a pretty good polygenic score. And there's arthritis in my family, and I'm starting to get arthritis, and for you young people, you should know that it comes up in these arthritic flares wherever you injure yourself. So I don't have any cartilage in my knee, and I don't only have one ligament, and I broke my finger, broke my toe, and that is exactly where I'm getting this arthritic, sort of flares that come up. So if I had known this early in life, the only sports I did were bad for your joints. I did volleyball, sideways jumping if you weigh 250 pounds, not a good idea. Basketball is jumping, skiing. And if I had known about this, I might have done what the English are very good at, sitting down sports. Biking, sailing, that sort of thing. So I think it's at that low-tech level, I believe that, and prevention and awareness, I think, that is going to make this useful. So those are just a sampling of medical polygenic scores. And for the behavioral ones, I'll give you the biggies. Bipolar, I have a very low risk, and kind of makes sense, there's no bipolar in my family. But this makes the point that, is that necessarily good? People call these risk scores, they're only concerned about risk. So is a very low score, does that just mean I have low risk for it? Or does it mean, it could mean something different? It could mean, for example, that people at the low end are flat in affect. They don't smell the roses. We don't know because nobody studies this other end of the extreme, but polygenic scores, I should have mentioned this earlier, are always normally distributed. So, that's the central limit theorem of probability, that it's like flipping coins, right? You flip coins, you count the number of heads and tails, you get a perfectly normal distribution. Here we're flipping alleles, it's the same thing. So these polygenic scores are always normally distributed, which means they have a low end as well as a high end. And we don't know much about that low end. Depression, I'm also low, and I don't have depression, bipolar in my family, but as you'll see, that doesn't necessarily mean much. ADHD, attention deficit hyperactivity disorder, it's not a very strong polygenic score yet, but it's interesting I'm in the middle, because my parents always said when I was growing up in the Catholic school system of Chicago, where you had to sit in these chairs that are lined up and you don't move for hours on end, they would say, if they hadn't invented the diagnosis ADHD, but I would have had it for sure, because I was always getting in trouble because I was too overly active. But the major component of ADHD, I think, is attention. And I always had the attention. It's just I couldn't stand being constrained for that long, in that way. So that may be where it's coming from, because this diagnosis combines those two things, hyperactivity and attention. Now here's the one that really surprised me, is schizophrenia, very high polygenic score for schizophrenia. And this is a good way of talking about, well, what if you find out things you don't want to know? And I think this could be practical. Do you know the research at the Institute of Psychiatry that I think is pretty good now, showing that the new forms of marijuana in the high, very, very high THC content is really a pretty good way of bringing out your genetic propensity for schizophrenia. And so I was never that interested in doing that anyway, but this would make me say, well, this is another good reason to avoid that. But there's a lot to think about here. There's also some work that suggests that people are more creative, possibly. But there is no schizophrenia in my family. and that I know of. But that makes the important point that 90%, despite the high heritability of schizophrenia, something like 70%, only 1 out of 10 schizophrenics have a first-degree relative who's schizophrenic. 90% of the time, they're not. They have no first-degree relative. So the important thing about polygenic scores is they predict at an individual level, not just at a family level. And this is the educational achievement score, which I'm pleased to see I'm very high. So here, the big one is Alzheimer's and dementia. You know, the genome, those 2001 whole genome sequences I showed you in Science and Nature. One is Craig Venter, an entrepreneurial person who was competing with the government sequencing project, and James Watson gave his DNA for that. Both of these guys said they didn't want to know about the one predictor of late onset Alzheimer's, apolipoprotein E. If you have 2 of the E4, it's just the name of the allele, you have a 15-fold greater risk of having Alzheimer's. And so, because there's nothing we can do about it now, they chose not to know. Well, I think people really differ on whether they want to know. I generally want to know about these things, so I decided I would know what it is. And it's a bit of a cheat in a way, because only 1% of the population are E4, so I had a pretty good chance that I wasn't going to have that. But what would I have done if I found out I did have a high risk for Alzheimer's, a high polygenic score? Well, even though you can't do anything about it now, by having these polygenic scores and being able to predict, I believe research is going to be able to do better at predicting, at preventing, because that's what it's got to be about, preventive medicine, rather than trying to cure these things once they occur. So I think there's that. And then also just at a practical level, I would want to know about this, because I would plan for it. I would plan financially, plan in terms of my kids. So I know there's differences on that, but I chose to know, and fortunately, not just the ApoE4, but there's a whole polygenic score for Alzheimer's disease, and substantially below the mean for that. Okay, so I'm sure we'll come back and talk about that, but I do think polygenic scores are going to be driven by people wanting to understand themselves and their children. And I know there's big ethical issues around that, so maybe we can discuss that later. I'm excited about the possibilities of polygenic scores transforming clinical work. All we have now are symptoms. We don't have any psychiatric disorder diagnosed on the basis of causes. So Alzheimer's, you can only talk about those brain tangles in a post-mortem. So you can't predict a disorder with anything like causes, but polygenic scores are causal. One difference that I think is going to be, it's already having an effect, is we're going to move from qualitative diagnoses to recognizing quantitative dimensions. As I said, polygenic scores are always normally distributed. So if this is the polygenic score for schizophrenia, there's no breakpoint here. There's no genes for schizophrenia. There are thousands of genes, SNPs, DNA variants, that we all have. And it's purely quantitative. It's how many of them you have. Then we draw a line. But we shouldn't pretend that there's anything etiologically distinct. Disorders are merely the quantitative extreme of normal quantitative variation. So I really believe, and I have a paper in Nature Reviews Genetics called Common Disorders Are Common Traits. There are no common disorders, just quantitative traits. So all this time we spend worrying about, do you have to have 2 of these symptoms for 3 months or 1 of these for 4 months? It just seems to me all a nonsense, that it's all quantitative. There are no diagnoses or disorders. And if there are no disorders, then there are no cures for a disorder. And we have to move towards prediction and prevention, rather than thinking someone has a disorder and you've got to cure that disorder. What we've got to do is ameliorate symptoms. A lot of the work in schizophrenia is not trying to cure people of schizophrenia. It's trying to help them, through cognitive behavior training, deal with their symptoms better. And it's very effective, but it doesn't cure it. And people are beginning to look at prediction and prevention. Instead of one-size-fits-all treatment, we'll be thinking more about individually tailored interventions. The big buzzword in the States is precision medicine. And related to what I said earlier, we'll switch from negative genomics, looking for genetic effects that cause problems, to positive genomics. That is, people talk about polygenic scores as polygenic risk scores, as if all they're doing is increasing your risk for disorders. But what about this other end of the normal distribution? We really don't know much about it. And I'm excited about the possibilities of getting away from all the negative and beginning to look more at the positive end of things. Because from a public health point of view, you might do better for a population to promote the positive rather than try to fix the negative. Long story there as well. So I'll end just by telling you about some research examples. And these are research from my area. These are classic questions we've always dealt with, multivariate issues. To what extent do genes that affect one trait also affect another? So this polygenic score for educational attainment, years of education, is the best predictor, genetic predictor we have of reading, as well as of IQ, intelligence. And a developmental question we ask is, if you find these polygenic scores in adults, how early can you see their effects in childhood? Genes generally account for stability. The environment tends to account for change from age to age. This polygenic score doesn't just predict GCSE scores or adult educational attainment. They actually predict 3% of the variance in first grade achievement, as assessed on national curriculum tests in England. The third classic way of going beyond just asking is something heritable is to study GE interplay. And here we look for, believe it or not, genetic effects on environmental measures. Most of the environmental measures we use, like life events or social support, show heritable influence, and that's because we're not really studying the environment out there. We're studying our experiences in using the environment. Like what goes on to stress measures is stuff like getting in financial difficulty, having conflicts with people. Those aren't the environment out there stressing you. They're stuff that you do, and that's where genetics comes in. And you can also show this at the DNA level. But rather than just studying the old questions, you can use DNA, polygenic scores, to ask about novel questions, which you couldn't ask about with twin and adoption studies. Like here's one that's is I think going to create some waves. If you look at private schools versus state schools in England, there's a whole grade GCSE difference, a standard deviation difference, big difference. And private schools assume that that's due to value they added. But actually it's not. That genetics is accounting for most of that difference, which you can study with DNA. It's mostly about selection. Private schools that select on the basis of prior achievement, ability to some extent, and if you correct for those things, there's no added value of the difference in GCSE scores goes to 0. Now, there's lots of other reasons why you might send your kids to a private school. I think they're mostly wrong too, but I think this is really important to recognize that genetics can account for some of these differences. I'm particularly interested in education. So we have a paper that came out recently showing that this DNA polygenic score mirrors intergenerational educational mobility. And the bottom line of this is that intergenerational mobility is the idea that university parents, you have kids who are more likely to go to university, but some of those kids don't go to university. The kids that don't go to university have lower polygenic scores. Conversely, the parents who don't go to university and have kids who go to university, upward mobility, have higher polygenic scores. And this is an example of how genetics is an index of educational opportunity and mobility. Lots to talk about there. And the third example, the last example, is looking at abrupt and massive social change. I have a graduate student who's Estonian, and she was really quite interested in the transition to independence in, when was that, 1991, 2001? And what she shows is that this polygenic score predicts twice as much variance in socioeconomic status in Estonia after the fall of the Soviet Empire. And so as you move from communism to capitalism, genetics becomes more important, which is the idea that opportunity allows people to fulfill their genetic propensities so that heritability can go up, which is why I say heritability can be an index of social equality. Okay, so that was a quick run-through of some of these applications, understanding ourselves, psychology and psychiatry, how it's changing psychological research already. And this is what I talked about today. I have a book coming out next year that it talks about sort of quantitative genetics, twin and adoption study and implications. And then the last half of the book is about DNA. And what I talked to you about today is kind of a preview of some of these chapters on predictive power of polygenic scores and personal genomics. The main message is that DNA revolution has arrived, and that it should be viewed as an opportunity for behavioral scientists to add a genetic dimension to their research. You don't have to become a geneticist. You can use this as just another variable. And you got to get DNA, genotype it, and analyze it, but it's easy to collaborate. Most universities have genotyping facilities. Cambridge is top-notch for that. There's still an issue of analysis for polygenic scores, but those pipelines are being worked out, so this will be simplified. But all you got to do is get DNA once, SNP chip once, and you get access to all these hundreds of thousands of polygenic scores. You can do this with companies as well. 23andMe has just launched. I have nothing to do with 23andMe. In fact, most researchers don't like them particularly. But they will actually do the whole thing for you. You can collect DNA from your subjects. They'll send you back the data. So this is, and there are these economies of scale. But the main message of all is that I really want behavioral scientists to join the DNA revolution. I was amused to hear that clinical psychologists used to use the acronym DNA to stand for did not attend. And what's really important is that we do attend and realize DNA is deoxyribonucleic acid. And the main message of all is that genetics is far too important to leave to geneticists. So thank you.